Well, first of all, I want to thank you guys for having me here today. It's an honor to be here, and I'm a stronger believer that science belongs to all of us. And part of what I have dedicated my career is to really point out to that point that it's not science happening in the lab. Everyone can be a scientist. So with that being said, the story I'm going to tell you today is a story of the model organisms. The things that you're going to hear today is this word constantly: invertebrate, flies, worms. What are those? So to break down and to give you guidelines of how we will be the afternoon, we will be learning what is a model organism, some examples of questions that these model organisms have solved in science. I'm going to tell you a brief story of part of my PhD work. I did my PhD in neuroscience with flies. And then at the end of the talk, I'm going to share a little bit of a story that the flies took me to Latin America to also expand the community of science down there, and how these animals can have a greater impact for economies that are underdeveloped. Can you hear me well? Yeah. Excellent. So I'm curious to hear from you a little bit what brought you here to learn and understand a model organism. So if one of you could sort of tell me, so I get a feeling of why you are here and why a model organism is interested to you. So let's start with the first question. So what is a model organism? So model organisms are these small animals. You can talk about a model organism as a yeast, a worm, which in the lab is called the C. elegans, a fly, a fish, and also the mice. And any organism that today can be genetically manipulated is a model organism. And for the purpose of time and for sort of focusing attention to you in the talk, I'm going to be focusing mostly in work that has been done in the worm and in the fly. The conceptual basics of using a model organism are pretty conceptually explained here. So imagine these boxes being a system. It could be your Twitter, it could be communities, it could be houses, it could be cells in your body. How do really I understand how the box red communicate or integrate information with the black box? How do I really know that those things are connected, and how much do I know in that complex environment how those two things are communicating? Pretty much the way we think about model organism is that they're a really simple way. You just have simple connections. You have the red with the blacks, that we can understand pretty well what happened between box red to box black. And then all that information that we learn in that simple system can be transferred in order to understand the complex. So an example for the further line, and I want you to keep in mind, is that if we do understand a problem in a fly, that problem and that solution has to be transferred back into a human, more complex problem. So one pioneer and a big discovery that lead out today for answering many, many questions, for example, about cancer, is Dr. Hurwitz. Dr. Hurwitz worked in MIT, and he was really passionate about understanding the cell death mechanism. And that means that some of your cells in your body are dying, and then new ones are emerging constantly. But to really understand why that happened, you really need to understand the molecular mechanism that are happening inside your cells. So what the observation that Dr. Hurwitz found is that when he was using the worm, now you're seeing the worm in a more early development stage, where is the egg, some of those cells that this animal had weren't dying. So the way he discovered that is he discovered the path, which is called the set path, and then you don't really need to memorize the names of those genes. But those genes are pretty important for the mechanism of cell death. How do you transform that basic discovery or fundamental discovery into what we know today about cell death and why cell death is important? Well, we know that, for example, in mammalians and even in humans, the mechanism of cell death is a little bit more complex, but I have color coded here the genes that are working in the worm that are exactly the same that are working in the mammalian and even in your human genomes. So there is a certain homology. They're not exactly the same 1, 2, 3, 4, 5 thing, but there is a certain homology on the form and the structure and the sequence that make those genes that make them homologous and identical. So you will see a little bit more of that. So from simple discoveries, this can take further in mammalian system to understand the whole new mechanism of cell death. Another classic example that is pretty remarkable, so about 100 years ago, Mr. Morgan was in Columbia University, and he was working with flies. He discovered that Drosophila was a really cool system to understand biological questions. And he will take classes like this. So these are how you work with the flies in the lab. And he will pretty much put them in boxes, irradiate them, and then wait for the next generation of flies and see how those flies could be mutated. That means some of the genes of this early generation will have changed to the next generation. So he will take fly by fly and see if he could observe any difference in the look of the fly. So that's called the phenotype. And he discovered one fly, and in this case I'm showing you half and half because genetics allowed you to do that. So this fly didn't have wings. And that was the primary discovery of the gene wingless. And flies have wingless, and you and I have wingless. It's a gene, and it's many, many years after, and now we know that the gene wingless, it's pretty important for a mechanism that is highly, now in the mammalian system is called beta-catenin, but the same protein that is in the cells that is called wingless is also in the human. And those proteins are the things that companies use in order to understand how can you make medicines to solve questions like cancer, leukemia, and so on. Another example that is a little bit more disturbing, but at the same time pretty remarkable, is that within the fly has a beautiful eye, and then you can use that eye in order to test that process of growth of the cell. And this is a person with, it's called the disease is tuberous sclerosis. Forgive my pronunciation there. But it's, in humans, what it causes is that you get abnormal growth of cells all over your body, and especially in the face of these patients. What happens when you have the lack of the gene TC1 is exactly the same. And what it has been found in humans is that when these humans have modifications in the same gene TC1 or TC2, they are the responsible for causing that disease in human bodies. So things that we have learned in the fly has been able to transfer into what now can we solve for this patient that has a mutation in the TC1 gene. As you say in your introduction, what are things that I can study in a fly, and what are things that I cannot study in a fly? And you always, when you ever design any experiment, you need to keep in mind that not everything can be solved with these little heroes, but many things at the level of molecular can be solved in that space. So I'm not going to study flies if I'm trying to solve conscious or psychological problems. However, flies are heavily used to understand memory formation. I cannot study the immune system in a fly because the fly keeps a pretty under-conserved mechanism of immunology. So that's why people use the mouse for, as a system for lymphoid system. And then programmed cell death has been pretty much discovered and explained by the use of these worms organisms. So in this part, I will encourage you to ask me some questions, but pretty much to summarize, the three big concepts I wanted to transfer to you here, it was that first, the model organism is really a tool to study and understand biology. The genes that have been discovered in these animals have been genes that today are important for the solving of health diseases. And then you really need to understand what problem in biology you are studying in order to use the right organism to solve the question. So normally, actually they haven't solved it. It's the insulin pathway in that case. And the TC gene is part of that pathway. So normally what you do when you know what gene got affected in any given case, you study how could you alter or stop the connection between the genes. So you really need to understand the structure of that particle and then see if the medicine pretty much is something that does this lock, and then don't let them act. Because here you have a mutation, you need to understand what was the physiological cause of not having the gene, and then maybe solve that part. So that will be the answer to that. But it's a general approach more than specifically for that particular case. Yeah, I can tell you my personal experience because it's the one I know. And how did I decide that I was going to study my biological question in flies, for example. The first time I saw a talk with these animals, and I was able to see how this professor was able to specifically track one cell in the brain from one place to another and do it 100 times and be able to say that's genetically the same cell, allowed me to decide that's the system I want to study, because if I do want to understand genes, it's a really powerful system for me to make claims about genes. Sleep disorders are also studied in flies, because believe it or not, the genes that we maintain for circadian rhythms were discovered in these little ones. So the circ and the purr, which is something things that keep you up in the night and in the day, keep me up in the night, are also found in the Drosophila and even in butterflies. A lot of the studies that we know for sleep disorders came from studies in the butterflies. So pretty much if the gene is conserved, you could think about studying. If you're studying physiology that involves more complex fluids, no. If you're doing biochemistry, I will go to a mouse because it's bigger, you can do more things, but that will be the way to sort of solve it. I agree. I completely agree with you that the path that takes from studying in a simple organism to go to complex needs people, needs discussion, and needs communication among the scientists, because it's scientists that are not doing the fly work and they're doing the human or the clinical work. And those need to be well known of what's happening in the early stages and be alert. Hey, this guy discovered something in the fly. Should we know about it or not? So it's a lot about human communication in making the leap. It's also about the communication of the fly community to tell people. And there is, for example, websites that say, hey, we found this gene. It might be affecting such and such disease. So I agree with you. It takes more people than molecules to say, yes, this is the fact. So I told you that I was going to talk today about small brains, and I was going to use these animals, right? So the key question is, do these animals have brains? Have you ever wondered if a fly has a brain? And the answer is yes. I mean, yes and no. In the case of the C. elegans, which is a worm, one of the which are here, and you can come and see in the intermission, one of the things that has been done for the C. elegans is that all the neurons in that 1 millimeter animal has been mapped. So pretty much in the tip of that worm, you have all the neurons that you can study, and those are neurons. In the case of the fly, the number of neurons that a single fly has is in the range of billions. So they also have a complex brain to be studied. So simple, but not that simple. The beauty of this brain is that inside that brain, we have a lot of genetical information. And for example, these colors are color codes of certain genes that are expressed in certain regions of the brain. So with that in mind, when in neuroscience we try to understand the brain, pretty much the approach we do, one of the approaches, is that the brain is full of neurons, and these neurons are all interconnected. There is heavy investment today, for example, in the United States for the project is called The Brain, which is pretty much to understand all the connections that are going in the brain, and with the hope that if we know the connections, we will know a little bit more of how do we react. Each of those, so for example, in this case, the blue and the brown represent two different collections of neurons. And then the way that these neurons connect from each other is called the synapse. So there is a single unit, which is called the synapse, that happens between two neurons, and they connect in several places. But that at this unique unit, there is a lot of information happening from the two cells. And then the one that I'm going to be talking to you today is also a connection that happens between the neuron that is in your brain that connects you down to your muscles. So whenever you are flexing an arm, and that motor neuron is telling the muscle to flex or relax. And that's called the unit of the neuromuscular junction. So it's a neuron to a muscle. All the activity that happens here is highly concerned of what's happening in your brain. And I will explain a little bit more detail what I mean with conserve, because it's a really important concept. For example, flies have 77% of the genes of one single fly are conserved in the ones that we know for human diseases. So a gene is pretty much these letters of A, C, G, T. And then when we say there is an homology, that means that the T is the same as the T, but in this case, the T is different than the A. But at the end of the day, the human and the fly genome are highly conserved in many of the regions where the genes get made. So the assumption, and again, is an assumption, is that the findings that I found in a synapse because of the genes that are happening in the connection of the brain of the fly are conserved and pretty much similar to what is happening in the connection of the human brain. There is classical examples. For example, if you think in two neurons connecting, in order for this one to find the target, it needs a protein and telling, go and meet here. That's called the cell adhesion. And the same protein that is used for the fly is the same one that we use in our own brain. There is genes that are in the fly that are highly diverse, and we only have one, which is kind of counterintuitive, but it happens too. So there is exceptions to the rule too. So when I say genetics and a practical visual for you to understand how can she modify only one gene in one animal. This is a classical example, and it's something that we see in the lab. So on the top is the white eye mutant. And I was going to ask you what is the difference between the top and the bottom fly, but I think I already said it. The bottom one has a gene that has been inserted. And because the gene was inserted, now the eye looks red. So we can really follow genes by the color of the eye, and there's many other markers. But because that gene has been inserted, it has mutated the gene white, which is for the color of the eye of the fly. So then you have a normal versus a mutant fly. So one gene has been modified, so it's the mutant for that particular gene. The other advantage, that a practical advantage for the work is exemplified here. One fly takes 10 days to go from an embryo to adult, and it takes one single female can produce in the average of 100 or 200 eggs. So if you are going to study one gene, that means you have 200 samples in these tubes to do your experiments. The other thing is that the genetics of putting things in and out are pretty sophisticated and well known up to now. So I really can do experiments when I decide the gene in that disease was conserved. That means that's similar. I want to know if it's in the fly, what will I observe in my particular biological consequence? And then I say, OK, I know that. Now I know the piece of the genome. I'm taking it out. And then by the marker, I can say, OK, now I put it in back. I mean, I know it's hard to see, but I used to do it every day. And it would take me 3 months to make a new mutant, for example. How do they grow? And the fly has a life cycle. They go from embryo to larvae to pre-pupae and pupa. They mate. So also the behavior of mating is well understood. And the genes that make that male mate are conserved in you guys too. So there is a lot of genes that also are conserved in behaviors. For my PhD, I decided to switch a little bit, and then was the inspiration part, right? I decided to focus in the larvae. Instead to use the brain, I decided to use the larvae. The reason to use the larvae was pretty much practical. The larvae also has a brain, and it's the brain in development, so imagine this baby now growing. And the circuits in that brain are really simple. And then it's a neuromuscular junction because it's one neuron connecting to one muscle, and I was able to see over and over the same synaptic connection between a motor neuron and a muscle cell. What happened if, and then in the intermission you can take a look, and some of you will say how tiny it is, and you have to do all this under microscopes. What happened if I put them under a microscope and I stain for a certain marker that is going to allow me to see their muscles? They're pretty remarkable. They're pretty beautiful. And in this case, for example, I'm staining with a marker for muscle. And when you open this larvae in half with a scissors, it has a map. It's like a Google map. So every segment, so the A1 to A6, is look of a region that has been connected by one single motor neuron. So every time I decide to ask a question for one particular gene, I can go to my region, which is that box there, and I can ask, how does the synapse looks in normal versus mutant condition? This is when you put the neurons on overlap, and that's how you can see the circuit, and this is putting you under the microscope. And then those red things are the synaptic connections that are making the interaction between the motor neuron and the muscle. If you make it a zoom of that, this is how a synapse looks under the microscope. Pretty much, in this case, I'm labeling the neurons with red and the muscle membranes and interactions in green. So every time I will decide or make experiments, I will count things like how many of connections this animal made. So I will count each of those circles and say 100. OK, if I took the gene out, how many will the animal make? And then I would say more or less. So that will give me a sense of growth of connections. So that was pretty much when I entered the lab. And it was heavy. I mean, at this point, a lot of scientists in the world are using this system to understand the basics of the connection between synapses, in this case, the neuromuscular junction. But we were also, in the time, wondering the question. And this goes back to, does science have a logic of why you do the experiments? Really, no. I mean, scientists are kind of in this wondering mode. And you say, hey, what about if? And my what about if was, this looked pretty static to me. I don't think, our system is static. You made a connection, you keep the connection, you stay with that. If you think that you're making memories or forgetting things, will that your brain be so static? So with that question in mind, I decided, because I could label each single neuron with markers and put them in the microscope, I decided to look at them in live conditions. So if I put the animal live under a microscope, what would I see? And trust me, I spent hours under the microscope looking at this. But pretty much the things I observed, so this is the same synapse. The first observation happens in 0 hours. And then this is the same synapse then a day later. So I will put these animals down, save them, and then put it back. Things that I observed is that we were in the lab wondering, do you see that sort of ball out there and it's not really connected to anything? There were many things that were sort of evolving and growing. So it was a constant growth. And that wasn't a surprise, because when I was counting connections, we knew from previous research that as the muscle grows, you make more connections because you have a larger surface in order to maintain the force for the muscle to move. So creating new connections was something that was an assumption. I will have more connections because it needs more power. But the surprise to me and my colleagues was that those two, for example, in orange up there, completely disappear in the 24 hours. So that starts sort of leading to a question where, do I make connections and then I delete connections? There is a machinery or a cleaner or an eraser. And I can tell you that's not an artifact. It was really constantly happening. Every time I will go back, things were growing and disappearing. So I decided to go even further and say, well, maybe this is too long of a distance. I want to know what happened in a smaller period of time. And pretty much what I found is that every single hour, these connections weren't as clean, as perfect as I was observing in fixed preparation. They were pretty active. And I wish I can show you a video, but this was pretty tiny and pretty hard to make. But all the arrows that you can see here are new connections that are being made in that synapse. And then the animal knows what to keep, and animal knows what to delete. So pretty much I'm kind of showing you how you make your new connections. This is a second example. So this synapse was sort of extending there, and then that one particle became those two and disappeared. A long story short, because this is work for 4 years of work, we start with the question, what are the genes that could be taking a role here, and what are the mechanisms that can make synapses disappear? And one of the hypotheses went back to the same gene that I showed you in the worm that was responsible for the death of the cells. And that corpse is called the SERP corpse, and those are the cells that are dying. And this gene is called SED1 in the worm, and it's located in the surface of the cell, and it's this Pac-Man that is eating that cell death. So we knew that SED1 was responsible for engulfing and eating. And we say, well, A, what is the gene in the fly? B, can this gene be the responsible, among many others, of eating the synapses? So that was the hypothesis. And you can see that structurally, Draper looks exactly like SED1, but it's in a different organism. And this Draper in the human is called MEF2, and it looks exactly the same. So the hypothesis was, can this gene, now that is in the surface of cell, be responsible for the elimination of these synapses? So what I did is I look at wild-type samples, and I will label them neurons in red, muscles in green. And you can see that they're pretty, these varicosities, that I call them the synaptic buttons. But then when I will take out from the fly the gene Draper, this is what I found. That all the synapses were completely full of this garbage, neuronal material around, and these balls that we discovered later that we call it the ghost buttons. And the reason we call it the ghost buttons is because they were synapses that were made, but never reached the muscle and never make the connection. So the muscle never responded to bound. And by not having the gene, make the synapse to sort of lose the, it's not aesthetic, but lose sort of the structure, that maintenance that you see, and also maintain these materials on top of the synapse. And if you think about those materials, those are synaptic connections. So they're still trying to find signals around. So the consequence of having that material is that, for example, if you look at an entire synapse on the top, is sort of the normal animal. In the mutant, you will see that those animals hatch a pretty remarkable less amount of connections. So that told me two things. One, Draper was responsible for the elimination of this. And second, the clearance of that material was important for the growth of those connections. So if you don't have that machinery that is sort of cleaning up the synapses, you could also affect the number of new connections that you can make in these circuits. In summary, what I have told you up to now is pretty much the research I did on my time in the PhD. And then is that I did other things while I was doing the PhD, but I think one of the things is that in order to understand the brain, the basic unit that neuroscientists use is the synapse. So that's an important concept. The second thing is that by using the genetics of these model organisms, we can really dissect out what can we learn later on in the brain. We pretty much set ideas for the rest of the scientists with the concept that connections that are being made in our circuits are not an static and on and off thing that are constantly happening. And that process now is called synaptic plasticity. And then ideas that you started, for example, a gene that was pretty much discovered for the function of cell death or maybe cancer was also having a function in how the synapses or in this organism are making connections. So that's also give you the complexity that biology has, that genes can be used in different type of problems and are solving different type of things inside of our organisms. So that goes back to genetics. And the female, you put a female with a male, and in the female you will carry half of the genes, and the male will carry half of the other genes. So let's say that the experiment is to introduce a gene. I will have gene X in the genome of my female. I will cross it to modify gene in the male. And that will give me a genotype that is XX modify. That will be one way. And then you just pretty much select in the following generations for something that looks like this, to make it simple. So then now,
how do you actually modify the gene itself? So how do you get that fly that has a modification?
I was wondering. So to make this mutant, there are several ways to do it. So the DNA is a chemical thing. So you can use X-ray, and that will break the A's and T's and C's, and make, in the next generation, we'll make an, let's say this. You will make an A, T, make a mistake, G, G, G. This is a mutant, because the X-ray caused this to change. So then the next generation is a mutant, and that's the mutant. There is also chemical modification, and in flies it's called EMS, which is a pretty nasty chemical, and we still do it, and we do it like this because it can modify us too. And with that, you do exactly the same. The genome and the generation of the babies are the ones that get modified, and then you just capture this visual. Yeah. You do the genetics of generations and also the modification in the next one, and start selectively picking the modified one by a marker. Yeah, and that's an important point because this modification has to happen in that mating in the first cellular division to transmit into the next, because it has to be in the ovaries of the next generation. That's an awesome question, and it's the magic of genetics, but it's not that magic. First of all, those chemicals reactions are random. So change can happen across the genome, and the selection is the key. So the design of how you select, for example, that red-white eye, is one way to detect if it has been inserted or not. Then there is a mapping process. So let's say this is your entire genome. In the mating, what you do is that if the mutation is here, you use animals that you know are modified in certain regions of the genome. And then when you find a match, because this had something and this now doesn't have it, then you know what in your new genome the modification has been made. In today's world, you send it to a sequencing machine, and you know what genes are affected. And it saves you a lot of time and money. Indeed, the worms were used as a prototype of what today we know of how do we sequence the genome of humans. So all the technologies that was developed for mapping genes in worms now is how you can send your cells and get your pretty much genome in your computer. Yeah, I was never the good one about the mathematical of that, but I think efficient is a relative number. I think the efficiency question also goes in the fact that one fly gives you 100 eggs. So then you have, and one mouse gives you 7, one human hopefully gives you 2 in 75 years. So the genetics happen faster and efficiently and less costly. So that's why I go with the concept of efficient being relative. So one of the things that I told you, and going back to the question of efficiency, is that, yeah, oh, sorry, I need to. So one of the questions that we had about efficiency, for example, is space and resources, and how much money does it really mean to do these experiments, and how this system could be an interesting way to sort of be used for economies that are underdeveloped. So one of the things we did as an exercise, it was to think in the 4 organisms compared to human and count the number of days, for example, that it takes to make one animal. So in average, for example, a fly will take 10 days, compared to make a new human will take you 25 years, hopefully. The fly will make 250 in those 10 days. The human will make 1 to 2, hopefully. And then the amount of time that takes to take 2 of you is from 10 hours in the worm to 75 years when you became a grandparent, right? So it's not pretty much efficient. But just for the C. elegans of the worm, there is also a space constraint, and that space is money. You need to have the space to maintain all these animals in facilities and time and personnel. So the fly and the worm became really interesting because you see, I mean, to maintain this tube, it takes a person to make that food. And you're going to see something here. and pretty much the passion and the curiosity to use it as a tool for understanding biology. So with that in mind, and also with my personal experience, I am originally from Chile. You will capture my accent. And when I came to the United States, it was, 13 years ago, pretty much coming from a small village down in Chile. And in Logan Airport, I realized that you guys speak English. That was a big haha moment. Oh, wow, another language. But I was really passionate about science, and science was my language. But I also knew that the resources that are available here to make the big discoveries are way higher than the ones that I had for me. So with that in mind, there is some statistics in terms of money. It's not just me sort of having a sense. So for example, in the United States, even though today is still it's not giving enough money to science, it's giving an average of 2.8% of money in GDP compared to science. If you think in countries like Brazil, 1.8. If you think in countries like Argentina, Chile, Colombia, Paraguay, they barely made the 2.5% of their GDP. And what makes United States great is that the investment that United States has made in science had made United States the great nation that it is. All the inventions and things have go down. I used to see United States as this amazing, cool, awesome country because you guys were doing the things. The reality is that if we compare only to the United States, US has 10 scientists working in a unit of 10,000 hours of labor. And in that unit, it's kind of weird, but China, for example, has 1.8 scientists. Countries like Chile, Venezuela has 0.2. That means that there's a really reduced amount of researchers that could do the science in those countries. And on top of that, there is not much investment for them to do their research. And the third remarkable statistic is that people like me, for example, that while I was in Chile, while we had really limited resources, we still were thinking and dreaming about science, and we were still be able to publish in the journals that people from other countries were publishing. And if you look at the publications from 2013, there is a remarkable amount of publications that these scientists still manage to put out for the public for the discoveries of the next generations. So with this statistic, in 2010, I convinced my professors, and I say, why don't we bring flies into the world of Latin America, and all the discoveries that 100 years have passed here can be transferred to people in Latin America, because with little amount of resources, they could discover the new molecules and the new genes that pretty much we could advance efficiently for what we have. So I named that course Small Brains, Big Ideas, and the intention was to seed the future of science in Latin America. So that's pretty much what happened down in the course. So we invited, so we have done this in 2010, in 2012, and now we're running one in 2014. So far, we have trained 60 students, and they are coming from countries like Argentina, Uruguay, Colombia, Brazil, Cuba. The person in Peru, for example, they didn't have a department of biology in his university. Now he started a lab with flies, and he has a department of biology. There is a student in Colombia that she couldn't make science because they didn't have the money in her lab, and she went back and used the worms, and now she's studying one of the pathways for cancer with worms in Colombia. And in 2012, she sent me 2 students to be part because she needed people to get trained. And now in 2014, we're doing the third version, and we're sort of creating this community of Latin Americans. So one of the
course.
Yes.
What's
So
the duration of the course?
it's 10 days. So we take all of them down there and the professors, and we make a cultural fest of science. One of the activities I made the students to go, and the way I make them to get excited about science, is to make a video. And that's sort of one of the ways they apply for the course. So one of the activities you saw. So I wanted to show you one of them, and his creative way to sort of show his. So I asked them to show their country, their lab, and the people in there, and be proud of what they're doing, so people can see them outside of Latin America. Yeah, so that's Guillermo. And you can see, I mean, the microscope is similar, one microscope for the entire university. And they take turns, but they still handle, and they get pretty excited of the things we do down there, because we give them new tools in order to do new type of experiments. And we also been able to sort of encourage to bring professors from big universities. So it's professors coming from Oxford University, and they volunteer to go to Chile. Professors from the medical school in UMass. And then they've been able to go and visit their labs and do small rotations and come into the United States for 2 or 3 months, which it makes a huge impact in what they're doing down in Argentina or in Chile. But it's also speed the process of the research they could do. So it's sort of a way to speed up the productivity that they can have in their own country, and to be an example for the younger generation of people who is locally in the country to become scientists. And then the last one I wanted to show you is, instead of me telling you, this is sort of the network that we have created so far. So we have students coming from different countries, and they are all spread in Latin America. Some of them has stayed working with the flies. Some of them has gone to England to do their further study and use another system. And some of them are now moms and daughters, I mean, have families, and sort of they are creating their own small network. And I think the last video I wanted to share with you was a little bit more of the impact of what American professors are thinking that we're doing down in Chile for these students. Yeah, so that's pretty much what I wanted to tell you about that, how this little fly could impact also the work of younger scientists and in an emerging economy. So I want to thank you for being here, for spending your afternoon with me, share my passion with these little organisms. I still, there is a lot of secrets that are hiding here that students can take advantage, and that's it from me. So thank you so much. We haven't gotten Mexican applicants, so that will be one. My networks in Mexico are not that extensive, so that's another one. So if you know somebody, please bring it in. Second, there is a certain amount of limited resources. So one of the things I do is convince the government of Chile, and that was to put money for this, and say, look, you are the ones who, you might not be the most developed, but you have the more stable economy, and you could be the hub for what's happening for the rest of the region. Today I got an email from a person from China who wants to go to the course in Chile, because in China they don't have this. So yeah, many things can happen to this, and I think we started small, but we can go big. So that will be the answer for that. Yeah, so I spent 2 years in MIT doing economy of science. The answer is not simple, and I think the approach is more, what is a scientist? What is the definition of a scientist? Is a scientist the person who is inside a lab doing the experiments, or science is a network? And you, as citizen, also can strongly participate, and the resources be distributed in a completely different manner. So if I tell you a country should have 10 scientists per 100, it's not a number that will represent the productivity of science that a country could have. But if a scientist has a strong network supporting him or her, even though if it's one, could be really powerful and strong. So I don't think it's a per capita number. It's about how much the science is able to network out of the laboratories. Yeah, my brain has been wired and unwired for English and Spanish. I could certainly do it in Spanish. It's funny that I learned science here in English, and there are many things I can not really do super easy in Spanish, even if it's my native language. But I will be happy to do this presentation in Spanish if needed.
Oh, no, I mean the videos.
The reason that I do the video and the course is in English in Chile. There are two reasons. One, the language of science is English. So if these kids in the future wants to become leaders in science, they have to learn from the beginning the rules of science. So I make a really strong commitment that they take that into consideration. Now, take me as an example. When I came here, I was finishing college. I didn't know, hello, my name is Yuly. That's all the English I had, with all the effort my father did to put education in my persona. I didn't know English, and I didn't have resources to learn English. So I'm pushing them, right? And it's kind of unfair, but I think they manage. I mean, when they get to talk the science language and the professors are there, they figured out the word in order to communicate. So that's why I say the course is going to be English. Yeah, the science is my new project. So I finished so the research I show you is research I did 2 years ago. So now other people is following up with that. The science is a competition, and it's now working in this network of science, and it's thinking it has one scientist and one fashion designer working together in order to translate scientific concepts into people. So now we're using a human necessity of you getting clothes covered, unless you go naked to the street, in order to figure it out that the world around you is science. So it's a year a competition. Right now we got 60 participants. I think 60 is my number. 61 participants from all over the world to collaborate with a fashion designer, and they are creating a garment, and we're going to have a fashion show by the end of September with concepts of science in fashion. So that's the science. And you can go to the website too. If you Google, you will find me. Yes, that's another strong limitation for countries like Latin America. So universities here pay for their students to have access to the scientific journals. So that's a reality even here, that if I'm not affiliated to a university, I do not have access to scientific journals. The countries in like Latin America don't have that, and you will be fascinated by the ability of Facebook to help these students. I constantly get emails from friends in Facebook. Hey, Yuly, does this journal is listed in the friend of your friend to your friend? Yes. And I shouldn't tape this, but that yes means, OK, here is it, and get it. And yeah, the journal doesn't get the money, but the science gets made. So yeah, I totally agree. We need a new, different system for that. Yeah. I mean, I started talk saying that my belief is that all of you are scientists. We were born curious, and the things that stay in us when we do the science is that constant and anxiety of curiosity for the new. But you were born like that. I mean, when you were babies, you were exploring the world constantly, and then we start making choices. With the fly, it's sort of a similar model, that things that now are happening in those countries couldn't have happened if they wouldn't have the training. For astronomy, it's way more expensive because you need the telescope, Chile, for example, is the country that has the best telescopes in the world. But they did a model where they share. Everyone can go. Because they culturally know that the sky belongs to everyone. So it's not that property thing or culturally it's mine. Because they're giving land and it's a beautiful sky, they get hours during the year that they can use as Chilean scientists. But everyone goes down there and they use it as an exchange for people to know about the country and to know about science. So these ones are easy because it's cheaper. So the cost investment in the early stage is cheaper, and you can create more people, going back to the number of necessary scientists in the society, pretty much faster than in the astronomy field.